Hello, my name is Meng, and I'm here to present my work on a prototype and model of a biological tri-state memory switch in mammalian cells. The biological motive of this project are biofilms. Biofilms are a complex community of bacteria within an excreted exopolysaccharide matrix, and they're the cause of up to 80% of infections in humans. There's a significant need for a method of preventing biofilm formation on medical devices that is local and dynamic. In this study, we use Copasi, a biochemical simulation and analysis software, to model a biological tri-state switch. This switch is part of a mammalian cell system that can destroy bacterial biofilms on implantable medical devices. So the whole entire system is comprised of two parts: the mammalian cell intaking a chemical stimulus and categorizing it based on a tri-state memory switch, and then using that categorization to take action, whether that be destroying biofilms or modifying cell population size. The scope of our project is focused solely on biofilm size and memory. So first, I designed my gene circuits. I then performed a literature review for rate constants and existing work before initializing gene and other subspecies concentrations. Then I created reactions and other species in Copasi, and I ran experiments with certain initialization conditions to verify the performance of my gene circuit. Finally, I fine-tuned parameters based on experimental results. So first, in the circuit design of three different subcircuits is the endoribonuclease system, and this is comprised of three ERNs, Cas-E, CSY4, and PSP-Cas13b, which represent the low, medium, and high memory states, respectively. So as you can see here, each gene, upstream of each gene is a constitutively expressed promoter, HEF1A, as well as two cut sites for the two other proteins. And the idea of this is cross-suppression, where as long as one protein has a higher concentration relative to the others, it's capable of suppressing the other two proteins by cutting the mRNA. And this allows that protein to establish a steady state concentration. Next is the max function and linearizer, and this relies on RTTA and doxycycline, or DOX. DOX is a proxy for bacterial quorum sensing molecules, and when DOX binds to RTTA, it's capable of activating TRE promoters. And as you can see here, these TRE promoters then produce CSY4 and PSP-Cas13b in a separate gene circuit. And the difference here is that CSY4 lacks the cut site from Cas-E, and PSP-Cas13b has no cut sites whatsoever. So this allows the higher states to dominate with steady state concentrations, despite there being a high concentration of a lower level protein already. Moreover, there's also a IYRC located on the PSP-Cas13b gene, and this slows down its translation speed relative to CSY4 by a factor of roughly 15. Finally, because, as you can see in the linearizer, we can only move up levels and not back down, the memory switch uses a similar tactic using ABA transcription factors as well as ABA to activate the PH1F promoter, and this floods the system with Cas-E in order to bring it back down to a low level memory state. As for my materials, the whole entire project was performed in silico, so I used Copasi, a biochemical simulation software, and on Copasi I was capable of initializing different species, setting up different reactions, defining global quantities, and eventually setting up time course trials, which allowed me to generate plots with respect to time. I had 7 experiments to verify my model. First was testing the cross-suppression of the proteins by initializing the concentration to be 0 for all of them. Next, I set 1 molar concentration for CSY4 and 0 for the rest to see if CSY4 could establish a steady state concentration. Third, I tested to make sure I could set or initialize Cas-E within the system through introducing ABA. After that, I tested whether or not ABA would be able to reestablish Cas-E if we had an initial concentration of CSY4. Fifth and sixth, I'm seeing if low quantity and high quantity DOX is capable of producing CSY4 and PSP-Cas13b as the dominant states respectively. And finally, test 7 is a combination of all of the previous tests, trying to see if I can establish Cas-E, then switch to CSY4 by introducing DOX, and finally re-establishing Cas-E when we want to. And as you can see, with initial concentrations, everything is cross-suppressed to a 0 steady-state concentration. When I have CSY4, it establishes a steady-state concentration and completely dominates over the other two proteins. When I introduce Cas-E at time equals 0, I see that Cas-E begins to dominate at around 6,000 seconds, and I basically initiate the low state concentration. When I have CSY4 already, and then I introduce Cas-E, as you can see here, Cas-E still prevails, so the memory switch works even in the presence of concentrations of other proteins. And looking at 5 and 6 respectively, when I input 1 molar and 60 molar DOX within the system, CSY4 and PSP-Cas13b are prevailing respectively. And finally, I can input ABA and have Cas-E dominate, and then I can introduce DOX for CSY4 to take place, and finally I can flood the system with ABA to reset the switch. So the model was indeed able to establish a stable tri-state memory in alignment with the hypothesized behavior of the system. However, there were some practical considerations to consider. Namely, IYRCs generally have a reduction of around 30%, and although initially I tried a factor of 4 decrease, or roughly 25%, that was not enough in order to effectively linearize between CSY4 and PSP-Cas13b, so I did have to switch to 15 times. Also, DOX required 60 molar for the high concentration to prevail. Obviously, both of these are not entirely practical, but that could also be due to the arbitrary assignment of my 0.1 molar for all of my initial gene concentrations, which could drastically inflate the required concentrations necessary to establish state changes. So next, I would definitely consider, because specific rate constants in the literature review is quite limited, I would definitely consider going to the lab so I can more accurately determine rate constants and just in general verify the practicality of the system in vitro. And this also involves introducing fluorescent reporter molecules within my gene circuits, so then I can verify through color that the proteins are indeed prevailing within the cell system. So in conclusion, our results do support the feasibility of the tri-state memory switch, and it can be applied to many scenarios. The driving force of this project was destroying and preventing formation of bacterial biofilms in medical devices, but especially seeing how some of the concentrations required are quite high, it could work even better in environmental and non-physiological situations, such as breaking down environmental toxins or developing biofilms to protect crops from parasitic infections. And again, future work is definitely warranted. Laboratory-based testing of the tri-state memory switch is definitely a must. And also, later on, I'll need to design cellular pathways for the downstream effects, or part 2 of the system, such as bacterial cell destruction and mammalian cell population growth modulation. So thank you so much for all of your time, and I hope you enjoyed my presentation.